My name's Lauren, and I work at Heritage Burial Services at Oxford Archaeology, and I'm one of the lead osteologists at Trinity Burial Ground. So my role day to day is managing the on-site laboratory where we are analysing the 1,500 skeletons that Steve mentioned in his talk earlier. Of all the skeletons in the burial ground, we are allowed to select 1,500 of those for full analysis, and I'm overseeing and undertaking some of that analysis as well. Why would we want to study archaeological burials? Well, there are a few very good reasons. For starters, we can look at the health and disease in the past. So obviously, quite a lot of our archaeological past, we did not have the advent of modern medicine, which we're lucky enough to have nowadays. So the patterns of health and disease that we see are a lot different to what we do now. And these very much change over time, right throughout prehistory and through the medieval period and so on and so forth. And so one of the things that we're wanting to look at Trinity is to look at the health and disease of people in the population of 19th century Hull. So we can also look at patterns in morbidity and mortality. This is looking at things like life expectancy, looking at the age that people are most likely to die at, looking at population composition, so proportions of males and females, adults or children. We can look at things like infant mortality rates. We know we're a lot higher in the past than they are now. So these are all things that we can start to investigate. And Steve already mentioned that one of the things that we're going to be doing at Trinity is looking at demographic patterns in the skeletal sample versus the demographic patterns that we're seeing across those burial registers and seeing if they match up or not. We can look at things like how well humans are adapted or not to their environment that they're living in. We can look at changes in funerary practices and how people's attitudes towards death have changed over time. Particularly in the Victorian period, funerary practices are very, very interesting. Harold might be able to talk about this a little bit more later on. And you've already seen some of the types of coffin fittings that we have from our site. And we can actually use these as well to help to date our burials. These are quite broad research topics. We can do a little bit more focused research, so we can look at people's diet and see the sorts of things that they were eating and what sort of effect that had on their health. We can look at how much people were moving around and migrating from place to place, and then also look into why they might have been moving or not moving, as the case may be. We can look at things like disability and care in the past and see what sort of conditions people were suffering from and what kind of help they might have needed with those things if they were having trouble day to day. And then we can also look at social interactions and things like interpersonal violence and conflict within populations. So these are just a few of the topics that we can kind of cover. And all of these things are things that we are going to be investigating in the Trinity population. In order to be able to find these things out, there's a number of things that osteologists like myself need to be able to record. And the photo that you can actually see on the slide is one of our osteologists, Vicky, in our main laboratory. And you can see she's got a skeleton laid out on her table there, and she's looking at some of the bones and she's recording them digitally on our database. So what we will typically do every time we have a skeleton in the laboratory for analysis is the first job is to get it out of its box, to lay it out on the table in an anatomical position, as you can see on the slide. And we will separate out any non-human elements first. So if we have any animal bones, because we do occasionally get little bits of those cropping up on the site, and we'll separate those out. We'll then make an inventory of what we actually have for each person, because they may be very, very complete. There may be some little bits missing, and we need to know these things. So we make an inventory of all the bones and the teeth that are present. We will then have a look at various bones and try to work out the age, death, and the sex of the individual. So that means we can try and determine whether they're male or female, and roughly how old they were when they died. We'll also have a really, really good look at their teeth and see how their oral health was, and see what kind of dental conditions they might have been suffering from, whether they had a lot of cavities, a lot of dental calculus, or anything else. We can look for something called non-metric traits, which we basically use to look at the normal skeletal variation within a population. Sometimes there are tiny little things you can see on a skeleton which are like inherited characteristics, and these are something that we can consider when we're starting to look within the grave stacks and see whether people may or may not be family members, so whether they're related or not. We will also measure some of the bones, so primarily the long bones, but because these skeletons are being reburied fairly rapidly, we're actually taking extra measurements on these as well for the legacy of the project and for archival purposes too. But we can use things like measurements of the femur, which is the thigh bone, to try and estimate how tall that person was when they were alive. And then one of the final things that we'll do is we'll give each skeleton a really good once-over, a really good look at it, and look for any evidence of skeletal pathology. And so this is what tells us things like whether that person was suffering from any diseases, whether maybe they've broken any bones, whether they had things like arthritis. And so we can start to learn a little bit about the health of that person. So basically, we're building up an osteobiography for each individual that we're looking at. You could probably imagine it's very, very detailed. Another thing that we're doing is we're starting to think about any further analysis for each of our skeletons. And so we do have a program of radiography, which we're undertaking. So if we want to investigate, say, a broken bone and try and learn a little bit more about how it broke, we can flag it up for X-ray. We've actually got our mobile radiography unit working on site today, so they're helping us out with that. And then we're also looking at selecting individuals for isotope DNA and pathogen DNA analysis. So the next slide that you're going to be able to see there is just a couple of little extracts from our human osteology database. So Oxford Archaeology have a bespoke online digital database that we record all the skeletons that we analyse in for all our different sites, and Trinity is no exception. So you can see the image on the left shows a summary information page from one of our skeletal records. That is just a summary page, and this will become populated as we add more information about a skeleton into the database. You can see the dark blue menu down the left-hand side. Each one of those little boxes is a different page of information that we'll fill out for each person. There's a completeness and preservation box. There's a couple of inventory boxes, depending on whether the skeleton's an adult or a juvenile. There's an age one and a sex one. There's dentition, and so on and so forth. So there's all these things that we can be filling in. And on the right, you can see there is a dental chart. And this is just an example of one of the sorts of forms that we'll be filling in for each one of those little blue menu boxes. So here you can see the upper and lower jaws represented, each one of the teeth that should be there, and each one of the tooth positions within the actual jaw where the tooth will erupt from. And if we click on any one of these teeth, we would see another form pop up, so we can record numerous bits of information about every single tooth for that person. So you can now hopefully start to imagine like how much detail we're going into. To give you an idea of how quickly we're kind of doing this, we're aiming to do sort of 1.5 to 2 skeletons a day each in terms of analysis. So that's how detailed we're going. So obviously the skeletons don't just, they're not magically excavated from the ground and they're all clean enough for us to look at. The tent is very muddy. You can probably also imagine with the amount of rain that we've had recently, conditions can get quite boggy in there. So what we have for the 1,500 skeletons that are undergoing full analysis is we have a process of cleaning and washing and drying those before they get to the laboratory, because the osteologists working in the lab need to be able to see the bone surface nice and clean so that we can examine it and gather all this information. Because each piece of information we gather, it's like a little clue to that person's life story. So what you can see here on the left is one of our processing staff, Bronwyn, and she's very, very carefully, in this case, washing some quite fragmentary juvenile remains over a sink. You can see she's using a little toothbrush. She's doing it all by hand. She's washing everything over a little sieve, so that she can make sure that she catches all the tiny little fragments of bone. I should stress that we're being very, very careful to keep all the individual fragments from a person all together. It's very, very important that we keep all the remains of one individual together for this entire process, and we're very carefully monitoring every time a skeleton gets moved from place to place across the site, whether that's from the tent to a storage cabin or into the washroom or somewhere else. We're making sure that we're making every effort to make sure that the remains of that person are kept together. So you can see that Bronwyn is washing here. She would then put all those pieces of bone into a tray that's lined with newspaper, and they will be taken to our drying room, which you can see on the right. We have several bays in our drying room where we can put these trays full of bones, which are very carefully marked with the number of each skeleton on them, and they will stay in that lovely warm drying room for about a couple of weeks. We do not want to dry the bones out too fast, because if they're subject to too high a temperature, then they might dry out too fast and they can crack and become damaged. So we want to keep them warm but not too hot. So they will dry in that room for a couple of weeks, at which point when they are dry, they will be put into clean bags and then put into clean boxes, and they'll be put into a storage cabin until one of the osteologists from the lab goes to pick them up and bring them to the main lab for analysis. You can see here, this is a still of in our main laboratory. We've actually got 3 laboratories on site. This is the main one, which is the biggest, and then we've got 2 smaller satellite labs, which are just across the corridor from this one. In the main lab, we actually have 5 benches like the one that you can see here. This is Mark, our other lead osteologist, and you can see that he's got the skeleton laid out in anatomical position, like in the shot that we had of Vicky earlier. And this is basically the place that the skeletons they'll have all their full analysis undertaken on them. The 2 smaller satellite labs have also got space for 2 people each to work in there. So we will have a total of 8 osteologists at any one time undertaking full analysis. And then the 9th bench is actually for doing a variety of other tasks. We're also collecting disarticulated bone from the site, and we're making an effort to quantify that as well. So that's what the 9th bench is used for quite a lot of the time. I should say, if anybody's not sure what disarticulated bone is, it's the bone that's been disturbed, so it's not in an anatomical position anymore. It's not really a burial, it's just sort of mixed up within the soil, or perhaps even within the grave, perhaps where it's been disturbed by a grave digger putting a later burial into a grave plot. And so we're collecting all these bones up as well, and we're making a concerted effort to count various skeletal elements so that we can work out how many people are also represented by this disturbed bone. For the rest of the presentation, I'm just going to go through a few little preliminary observations that we've made so far. So we have been analysing remains on site since the end of October. I last counted our numbers on Friday, and I can tell you that we'd analysed 916 skeletons by Friday. So obviously we've done a few more than that now. So we're barreling along. We're nearly 2/3 of the way through, which is great. And some of the things that we're finding, I can have a little chat about now. I should stress that these things may change because obviously we have another 600 skeletons to go. So these are only very preliminary patterns that we're picking up in the data. These things might change when we've got the full set to look at later on. But at the moment, it's looking like about 1/3 of the assemblage are actually children. And when I say children, I mean individuals under 18 years of age. I should say we do have a lot of very young children within that number as well. A lot of them are actually concentrated at the very eastern end of the burial ground. You can see an image from WebMap on the screen there. Now you can see the roundabout on the A63 road there, and our site just to the southwest of it. Now at the very eastern end of the site, in this sort of thin strip with all the purple blobs in it, we're getting a lot of very, very young children from there. So preterm babies, newborn or perhaps stillborn babies, and also infants up to about a year old. They're buried in quite close proximity to that mortuary building that Steve mentioned, and also like on the south side of the jail. Now this is very interesting because this finding corresponds with some historical evidence that we have for there being secretive burial of the very young in this area. There is actually historical evidence that people were burying small children in this area after the burial ground had gone out of use. Some council workers who were planting trees in the cemetery in the 1880s actually found 40 burials of very young children while they were doing these groundworks, and they were actually exhumed, and they were never identified, but they were buried somewhere else. So we did anticipate that we might find a lot of very young burials in that region, and that has actually been the case. Having said that, we do also have very young children burials in the rest of the cemetery. We just have this little concentration in that far eastern end. So that's something very interesting that we shall be pursuing the research. Another thing that we've found osteologically when we've been looking at the juvenile bones is that a lot of the child remains seem to have deficiency conditions. Now, the chief ones that we're seeing represented are scurvy, which is a vitamin C deficiency, and also rickets, which is a vitamin D deficiency. So the scurvy, which I didn't include a picture of the scurvy because the lesions are quite subtle. They're usually in and around the cranium and maybe on the shoulder blades, and they're quite difficult to photograph. And also, if you're not used to looking at scurvy lesions, they are very difficult to decide what you're actually looking at. So I thought these ones I'm showing you now would be a bit more obvious. The fact that we're getting a lot of scurvy in the very young suggests that children are not eating diets that have sufficient vitamin C, And when you have children that are vitamin deficient in terms of things like they're not eating enough fruit and vegetables, we might be more likely to see other deficiencies in their skeletons as well. Now, the vitamin D is slightly different to that because humans chiefly get their vitamin D from sunlight. So the fact that we've got a lot of children that are exhibiting rickets suggests that for some reason they're not getting enough sun. So it could be that maybe very young babies are being swaddled. Maybe they are being kept indoors a lot. Maybe they're wearing a lot of clothes. That means that they're not getting that sunlight exposure on their skin. Or it could mean that they're actually living and or working in conditions that are very dark. And what rickets actually does to the skeleton is, because vitamin D is very important in people who are growing and making your bones nice and strong, what happens is the bones get too soft. And then just everyday actions like walking around or even crawling in very young babies, crawling and putting normal body weight on things like legs, which you can see here, they become bowed and bent because the bones are literally bending under the pressure of the weight of that child's body. So you can see on the left, we've got a skeleton that's laid out, that's from Trinity, and we can see that the lower legs are very, very bent. And that's a close-up of the same skeleton just in the centre of the slide there. And then on the right, you can see an example of some children. This photo was obviously taken quite a while ago. This is a photograph from the Wellcome collection of 3 children who do have rickets, and you can see those same bending deformities in the legs. And then in the adult population, we're seeing a few different things. We are actually seeing rickets persisting into the adult population, so we get those bending deformities never actually go. So we are seeing people that are fully grown adults that have those residual bending deformities in their legs as well. But other things that we're seeing in the adult population include tuberculosis, which you can see in the image on the right. That is a spine where you can see the front part of the spine is up to the right side of the picture. And the little bits that are sticking out of each vertebrae are kind of the knobbly bits that you feel on your back if you run your finger down your own spine. So you can see that in this case, the lower spine has actually collapsed at the base and then it has bent forward. And this is caused by tuberculosis. We are seeing a lot more of this in the Trinity population than we were expecting for a population of this date. And interestingly, only quite a small proportion of people who have tuberculosis will actually end up with the skeletal lesions. So we can probably assume, based on this, that tuberculosis is actually quite rife within the population. Another thing that we're seeing as well is a high percentage of nasal fractures in the adult population. I should say in men and women, and I haven't checked the numbers for these, so I can't see if anybody is sustaining more or less of these than the other. All I can say at the moment is we've got a lot of broken noses. And although these can be caused by accidents, the chief reason for somebody having a broken nose, unfortunately, is being hit in the face. So make of that what you will, pending more analysis, I should say. We're also seeing evidence of a few quite rare conditions that we don't often see or see at all osteologically. But now, I should say that as well as the skeletons being excavated very carefully, we're also taking soil samples from certain regions of the skeleton to look for little extra things that might be too difficult to see just in the ground. So we take a little soil sample from around the skull, we take another one from the chest cavity, and we take another one from the inside of the pelvis. And then these get processed and sieved, and we have a little look around in the remains there once the soil's all washed off, and we see what's in there. And you can see a couple of the things on the slide that we've been finding in things like the soil samples. Now, we've been seeing a lot of evidence of calcified arteries, which is not something that we see very often at all archaeologically. The picture that I have of these calcified blood vessels on the slide is actually from a different site because I haven't had any properly photographed yet from this project, but they look exactly the same. And these are basically little hard calcifications that you get from inside of the arteries. They're caused by having an unhealthy diet or lifestyle, and they're most commonly associated with things like heart disease, and they can lead to people having angina or a heart attack. We're also seeing super rare cases of things like this other example that I have on the slide, where my colleague Abby was out in a tent assessing a skeleton a few weeks ago, and she noticed that there were these large balls of what looked like, I don't know, cauliflower or popcorn or something in the pelvic cavity of this skeleton. And we haven't actually had this skeleton through for full analysis yet. It'll still be drying in the drying room. But a cursory Google of these unusual popcorn-looking things suggests that this actually might be a really unusual type of malignant ovarian cancer. And I mean, nobody working in our laboratory has seen this before. So we're going to have to do a little bit more research on this. But I think this just gives you an idea of the sort of unusual things that we're seeing within this population. Hopefully you've enjoyed listening to a few of these preliminary observations that I've told you about today. Hopefully I'll be able to tell you a little bit more about what we've been finding in subsequent webinars if you'd like to join us for those. And that's about it for now. We'll be continuing our analyses for another few months, so probably until the end of August. And by that time, all the skeletons will have been reburied. So thank you very much for listening, everybody, and I look forward to your questions.